In the early years, Colombo Plan assistance from developed to developing countries comprised both transfer of physical capital and technology, as well as a strong component of skills development. Hence, while infrastructure by way of airports, roads, railways, dams, hospitals, fertilizer plants, cement factories, universities, and steel mills were constructed in member countries through Colombo Plan assistance, a large number of people were simultaneously trained to manage such infrastructure and the growing economies. Now, the key point here is what the motivation was for setting it up in the first place, the Colombo Plan, and that was to battle communist movements in their countries. And who were the communist movements? We're talking about, of course, Russia and China. And the big funding came from the United States, but it was the United States pouring money into Commonwealth countries, where they're talking about developed and developing countries. So here you can see that all of Australia, Britain, Canada, New Zealand were the developed countries, the developing countries as they call them. They also used to call them underdeveloped countries, for Ceylon, India, and Pakistan. Now, one thing that I've noted from the studies on fluoride is that fluoride poisoning was a recorded and documented problem in India in particular. Also in China and also in Japan. That's where fluorosis, what they called endemic fluorosis, was found, and had been described as being a particular problem in Ceylon. Now, to understand that, I'm going to just get onto Google Maps for a sec and show you a little bit of the geography. So here we have a map of the world, and that's where the Nuffield Foundation is based, United Kingdom. There's Sri Lanka down there with the little star. There's India. Now I'll just go a little bit closer in on India. And there have been fluoride poisoning is a problem in North India and South India. In Sri Lanka, the studies that my parents did showed that there were 3 areas of fluoride poisoning. One was here, inland from Trincomalee, which was a World War II command center and a transit point for the ships between Australia and the Middle East. So I had a lot of patients, World War II veterans, who, when they knew that I was from Sri Lanka, would say, "Oh yes, I've been to Sri Lanka," because they stopped over in Trincomalee on the way back from the war. And inland in here is a big area of fluoride poisoning, and that is characterized by extensive staining or mottling of the children's teeth and some fluoride bone deformities. See, fluoride poisoning causes deformities in the bones. So that's one area. The other area of fluoride deposit was over here. Third area was down here. Now that's inland from Hambantota and Tangor, which is, these are all ports in the south. But the big deposit of fluoride was in this area over here, which is Anuradhapura, and south of it Polonnaruwa is around here. The ancient Buddhist capitals of Sri Lanka. And the deposits of fluoride were around here. Now my father, or my parents, attributed this to it being the dry zone. And he talks about a division between the dry zone with lateritic soil and or the wet zone with lateritic soils and different soil types, and the dry zone on the eastern part, relating it to the monsoon. Now, it is, the reason for that is that in this area of Sri Lanka, the hill area, which is also closer to Colombo and also around the tea estates, there is no problem with fluoride poisoning. There's a very low incidence of fluoride. Now, the problem with the fluoride poisoning here is fluoride in well water. The irrigation tanks, or the irrigation reservoirs, did not have a big collection of fluoride, so it was something that was there within the soil. Now, in the studies, my father, my parents, said that some areas or some incidents of fluorosis could not be explained just by the water samples, and they didn't explain that incidence of poisoning. Now, there is another part to this story. That is just the fluoride studies. Now, first of all, I'll just show you the geography of where Kandy General Hospital, that's where the lab was. That's Kandy. And there were some pictures of Kandy General Hospital there. And Kandy was the ancient hill kingdom that was the last to resist the British and hold out against the British. This area around Kandy, there are extensive tea estates, and then all around the hill areas. Around these hills around Kandy, in the Hanthana tea estate in particular, my father had an arrangement with the people running the tea estate to do experiments on the tea estate workers to measure their hemoglobin. And he even started off trying to do bone marrow samples, which are very painful procedures, on these people who were working on the tea estates. So he'd arrive with his team, and when the workers were lining up for work, they would have a venipuncture, meaning someone would take blood from them. Initially, they tried to do bone marrows as well, but they found that there was a very high absenteeism rate, not surprisingly, considering that these people were paid something like $1 a week or something along those lines, a few rupees a day. They were paid a pittance. And the people who owned these tea estates back when this research started were the British, in particular, Liptons and Bushells. And it was in fact Thomas Lipton, who was a British mason, who brought in, he's the person who brought Indian slaves from South India. And he got these Indian slaves to walk, what they call indentured labor, or coolies. Got them to walk all the way here. Lots died on the way, I'm sure, but they weren't counted. And they were brought to work on the tea estates over here. Now, the British also transported Indian slaves all over the world. They transported them to Africa, they transported them to Fiji, over here, Fiji, to work on sugarcane plantations, and they also transported some to Queensland. And this was before they started kidnapping Pacific Islanders to work on the cane fields in Queensland. That was particularly in around Townsville. And Townsville is actually named after a slaver. Townsville is the person who brought what they call Kanaka laborers to work on the tea estates over here. But before that, they had tried an experiment with, yes, Indian tea estate workers, what they call indentured labor. Then they tried an experiment with Chinese coolies. That's what they call them, coolies. And this is all stuff that happened before the Federation of Australia, so before 1901. This happened in the 1800s when there were still separate states. There was a separate state of Queensland and New South Wales, Victoria, South Australia, and Western Australia. The Northern Territory was the Northern Territory of South Australia. Now the Northern Territory is administered from Canberra. So, to get a bigger picture of what was going on in the with the Colombo Plan, you look at the countries: England, Australia, New Zealand, or United Kingdom, Australia, New Zealand, Canada. Those are the developed countries. The developing countries, or underdeveloped countries, were Pakistan, India, and Sri Lanka. So what was going on during the Second World War is that the British power, the link between United Kingdom and Australia, was right there, Singapore. So the flights were through Singapore, and the shipping was through Trincomalee over here, and the link was with Western Australia and the port of Fremantle. So that was the power of the Indian Ocean during the Second World War. So it makes a lot of sense that they would have been dumping their fluoride waste somewhere, and it appears that they dumped it in the Third World. And you'd expect that they would dump it in Sri Lanka and India. Some would have been dumped by the Japanese in China, which explains the fluoride poisoning in China. Some was dumped in Japan. I wouldn't be surprised if there was some dumped in Korea as well. I wouldn't be surprised if some was dumped in Australia and some dumped in Africa. I would be very surprised if any was dumped anywhere near Buckingham Palace or London. Right, that's the way it was. So, the research on the tea estate workers is what I'm going to look at next, because the British had a very obvious interest in maximizing the efficiency of the slaves on the tea estate workers on the tea estates. And that explains the Nuffield Foundation's support of my father's work researching the efficiency and the vulnerability of tea estate workers. I have two of the papers that my father and his team published on, or had published, on anemia. This one is given to me by my father. You can say he's written with the compliments of the senior author. And then underneath, my mother has signed K. Senewiratne brackets JA. It stands for junior author. That's how, that's my father's attitude, my parents' attitude to this. This was accepted in 1973, accepted July 1973, and published in the British Journal of Nutrition in 1974. Vitamin B12 absorption in megaloblastic anemia. That is looking at vitamin deficiencies, vitamin B12 and folic acid deficiency caused by malnutrition. And this is called some problems in the management of anemia in tea estate workers in Sri Lanka, in the Journal of Tropical Medicine and Hygiene, August 1974. This is by the same authors, B. Senewiratne, J. Hettiarachchi, who was called Hetty, and Kamalini Senewiratne, my mother. Department of Medicine, University of Sri Lanka, Peradeniya, and Clinical Research Unit, General Hospital, Kandy, Sri Lanka. Now this paper is also about malnutrition. And it says, anemia, often of a severe degree, is common among tea estate workers in Sri Lanka. Here's a quote of his own paper. A large percentage of the anemic patients admitted to the female wards of the General Hospital Kandy were workers on tea estates, most of whom had been working almost up to the time of their admission to the hospital. Anemia could contribute to the mortality and morbidity of these workers and result in reduced work output. This is of serious economic importance since tea is the major export of this country. It's also of particular interest to the British tea companies, of course. A long-term study was therefore undertaken on a tea estate to determine the incidence, type, and severity of anemia among the workers and how it could be corrected. Now, an unselected group of 347 workers on the Hamthana tea estate, 2,500 feet above sea level in Kandy, were investigated. All the subjects had reported for work on the day of the study. The hemoglobin and packed cell volume were estimated and a blood film made. Where possible, a specimen of feces was examined for parasitic ova and cysts by a concentration technique which was a modification of Ritchie's formol ether method. Where hookworm ova were present, a quantitative estimation was done. Information regarding the diet, income, social history, laboratory facilities, and the use of footwear was obtained. The diet was classified into 4 categories in relation to the intake of animal protein: meat, fish, dried fish, eggs, adequate animal protein daily, moderately adequate protein, which is 3 to 6 times a week, inadequate 1 to 3 times a week, and 4 grossly inadequate, less than once a week. So he used the World Health Organization 1968 definition, and this has relevance because his boss, Max Rosenheim, was the boss of the World Health Organization at that time. As you can see in Max Rosenheim's obituary. Now, my father had another boss in England, and his name was John Walters. And here in his What I Taught and Learnt in Sri Lanka, my father, who has a tendency to drop names, drops the name of John Walters, who I just looked up for the first time today. And my father writes as to why he left Sri Lanka. He says, "Speaking for myself, I did not return to Ceylon in search of money. I did not turn down a consultant appointment in a major teaching hospital in London, complete with an already established Harley Street private practice of my boss, the late John Walters, senior physician, Hospital for Tropical Diseases, whose senior registrar I was, to return to some bankrupt dump looking for money. I knew it was financial suicide at an annual salary of 9,500 rupees, something I could learn in less than a week in London. I returned because I wanted to live and work in the country of my birth and among my people." So, who was John Walters? My father's making out that he could have got his you could say, inherited his practice. The thing is, medical practices don't work like that. This is a little bit about John Henry Walters. Yes, Lieutenant Colonel John Henry Walters served in Iraq and Egypt during the Second World War and worked with prisoners of war, which started an interest in the neurological effects of malnutrition. After the war, he worked in the Gambia at the Nutrition Research Station, then in Kuwait and Lagos. Towards the end of his career, he was a consultant to the London School of Tropical Medicine and Hygiene, and after his retirement returned to Devon, where he was Chief Medical Officer to the Newton Abbot steeplechase. Right. Now, he was another military doctor, along with a senior military doctor. He was a lieutenant colonel. And along with, and this is actually from the Wellcome Library. And so he's another person involved with the Wellcome Trust. And this is his collection. And it gives you an idea of the people who were behind the scenes. organizing my father's research. John Henry Walters, MD, MB, Beecher, FRCP, MRCS, born 1909, died in 1993. Lieutenant Colonel of the Indian Medical Service, Consultant Physician, Hospital for Tropical Diseases, London, and the Tropical Unit of the Queen Mary Hospital, Roehampton. And you see a connection here with the eugenics movement, and also these are the boxes. Clinical photographs, mainly malarial related conditions. Indian Medical Service reports on typhus, histoplasmosis, and beriberi. Beriberi is again related to malnutrition. And reports, case studies, and lecture notes on clinical diagnosis and sickle cell. Now, sickle cell trait, which is a hemoglobinopathy, results in resistance to malaria. So that's what they were interested in. Stopping malaria in the troops during the Second World War. Box 2 is clinical photographs, mainly of genetic diseases. Just like the Nazis, the British were very interested in genetic diseases. And box 3, glass lantern slides of clinical conditions. And it doesn't say a lot about him, but it says during the Second World War he saw active service in both Iraq and Egypt and worked with Far East prisoners of war after their release. The latter work led to an interest in the neurological consequences of prolonged malnutrition. Starvation, in other words. He studied starving people. This interest continued after the war from 1948 to 51. Walters worked with John Waterlow at the Nutrition Research Station in the Gambia, where they researched the effects of malnutrition on the livers of Gambian children. After 3 years working in Kuwait, he became medical officer in charge of the West African Council for Medical Research based in Lagos. In 1958, Walters returned to Britain to become consultant physician to the Hospital for Tropical Diseases, a post he held until his retirement in 1974. Now, custodial history, he was given the Wellcome Tropical Institute. It says in 1997. I don't understand what that means exactly, because in 1997 he was dead. But after he died, presumably his collection was given to the Wellcome Tropical Institute or the Wellcome Library. So he's another of the people involved in the Wellcome Trust. And the other thing that is particularly relevant is that immediately before he was my father's boss, he was studying the effects of malnutrition on the livers of Gambian children. Now, if you look at my father's research on anemia, firstly, it's obvious that this is not informed consent. It was an unselected group of 347 workers on the Hanthana tea estate. So when these workers were lining up for work, they were photographed and they were tested, investigated. They had no say in the matter. Now, one point that is worth reading over here is a detailed study of the workers who were found to be anemic was done over a period of 18 months. Since it was a long-term study, it was essential to get the cooperation of these apparently normal people. it was observed that a venipuncture resulted in a very high rate of absenteeism at subsequent examinations. It was therefore not possible to estimate the serum concentration of iron, folate, and vitamin B12, or to do bone marrow examinations. Elsewhere, he says that they did try doing bone marrow examinations early in the piece, but they were not able to persist with it because, as I said, as he says, of the high rates of absenteeism. Now, here you get to the crux of the matter. What was the anemia due to? Firstly, I'll read the summary. 36% of 347 workers on a tea estate were found to be anemic. Hematinics were given to 92 anemic subjects over a period of 18 months. All 69 subjects in whom it was possible to correct the anemia had an iron deficiency, either alone or combined with folate, vitamin B12, or both. The long-term control of hookworm infection and the improvement of the diet do not seem to be possible in this community for socio-economic reasons. That is the key point, isn't it? So he's saying they can't improve the diet or control the infection of these worms that was causing anemia as well, because of socio-economic reasons. Well, what about improving their socio-economic situation? Would be the obvious way of fixing it up, but my father was not pushing for that, because this was, after all, his objective was to be selling vitamin tablets and vitamin B12 injections and tablets of folate. So he writes, despite a moderate to severe anemia and the supply of free drugs, 25 of the subjects failed to take iron tablets regularly, indicating that long-term therapy with iron is unsatisfactory. Other methods of iron supplementation are discussed. Right. So he thanks the Nuffield Foundation for a grant to one of us, brackets BS. And they also had a grant from the National Science Council of Sri Lanka. Now here he says, despite the fact that none of the subjects were given antihelminthics. Antihelminthics are drugs against hookworm. In 69 of 74 subjects who took the iron tablets and did not have any disease which could produce an anemia, the anemia was corrected by iron supplements. This would suggest that the major cause of the iron deficiency is an inadequate intake of utilizable iron. In many countries where anemia and hookworm disease are prevalent, the dietary iron intake is high. However, the iron is mainly of vegetable origin, since the protein intake is low. It is known that iron from animal sources is better absorbed than iron in vegetables. 75% of our subjects ate a diet that was deficient in animal protein. Therefore, the most important factor in the management of anemia in these workers is the provision of an adequate diet. And this is the crux of it. Because of their very poor per capita income, average 10 rupees, 10.7 rupees a month, approximately half a pound a month, it is not possible for them to improve their diet. As a result, lifelong supplementation of the diet with iron is necessary. The simplest and cheapest method of supplying iron is by the regular supply of iron in tablet form. In our study, we found that this method was not very successful. Despite frequent visits to the estate and a series of talks on the importance of iron, 13.1% of the anemic subjects failed or refused to take the iron regularly for a long period. A further 14%, who had initial rise of hemoglobin on oral ferrous sulfate therapy, presumably stopped taking tablets after a few months, since the hemoglobin concentration had reached normal levels, subsequently dropped, or the subjects had to be given intramuscular iron, an injection in other words, to restore the hemoglobin concentration to normal levels. An alternative method of iron supplementation is by the parenteral administration of iron, either intramuscularly or intravenously. Repeated intramuscular or intravenous injections of iron are obviously impracticable for long-term therapy. A single large intravenous injection of iron dextran complex is a possibility. However, serious side effects have been reported, and the procedure has not been adequately evaluated. It is very expensive and impracticable on a large scale. A single intravenous injection of undiluted iron dextran complex is a more convenient alternative, but experience with this form of therapy is limited. The most suitable prophylactic method would be by the addition of iron to some item in the food that is consumed daily. The addition of iron to milk is unsuitable, since these workers did not take adequate amounts of milk. Flour would seem to be the most suitable vehicle. Cruz and Pimenta de Mello in 1948 have administered small doses of iron mixed with staple foods such as manioc flour. However, workers have claimed that the iron added to flour is not easily absorbed, possibly due to the high concentration of phytates and phosphates in flour. Further confirmation is necessary before this important finding is accepted. We have found that the addition of ferrous sulfate to flour has the limitations in that the concentration of over 60 milligrams of iron per 75 grams of flour produces a metallic taste, which made the food unpalatable. A trial is now in progress to investigate the biological availability of iron added to flour. Preliminary observations have shown that the rise of serum iron comparable to that with oral ferrous sulfate is obtained with iron that is added to flour. And this is the key point, of course, is the fact that he's saying that long-term control of hookworm infection and improvement of the diet are not possible for socioeconomic reasons. In other words, people who are suffering from severe malnutrition should not be given enough money to buy food. Instead, the tea estate is supposed to be supplying them with iron tablets. Now, it so happens that Wellcome Pharmaceuticals sold iron tablets. And the same is the case with the B12 absorption in megaloblastic anemia. This paper. Again, the megaloblastic anemia, which is a type of anemia where the cells become abnormally large. And here we are. This work was carried out by the Clinical Research Unit, which was set up by the Nuffield Foundation, London. We are grateful to the Nuffield Foundation for a generous grant to one of us, brackets BS, and Professor S.W. Bibile, University of Sri Lanka, and Professor G.M. Wilson, University of Glasgow, who have supported our work. Now, this Professor G.M. Wilson, as we saw, was a member of the British military, and so was Walters, and Max Rosenheim was the big boss. Now, the other side of the picture had to travel to Australia and find the career of Neil Hamilton Fairley. He was in Egypt as well, and an expert in tropical medicine in London. He returned to the Australian Army as Director of Medicine in his 40s, and he was for 5 years medical officer at the Bombay Bacteriological Laboratory. Now, he coordinated activities regarding malaria and promoted a new drug called atabrine. After the war, he returned to London and was consultant physician at the Hospital for Tropical Diseases and Wellcome Professor of Tropical Medicine at the London School of Tropical Medicine and Hygiene. Right. So Brigadier Neil Hamilton Fairley, he was made a commander of the military division of the Order of the British Empire, and he flew to Java in 1942. Java was then producing 90% of the world's quinine. So he flew to Java after Japan entered the war in 1942. And he tried to buy up the entire supply of quinine in Java. But of the two ships, he bought 120 tons of quinine. So rather than letting the quinine be used by everyone who needed quinine, the Australians tried buying it all up for the war effort. Not just the Australians, but the British, because the British, it was after all a British war effort. Now, the key thing with this malaria research is that the idea was to minimize casualties from malaria. They were trying to minimize their losses of their troops from malaria. They were not interested in the preventing or curing or stopping malaria amongst the civilians of these third world countries, not in the least bit. They weren't even interested in stopping malaria in civilians of their own countries, except where it impacted on their military agenda. And Neil Hamilton Fairley, he was the boss of Frank McFarlane Burnett at the Walter and Eliza Hall Institute. He's also the father of Gordon Hamilton Fairley. Gordon Hamilton Fairley is a professor of medical oncology, born and raised in Australia. He moved to the United Kingdom, where he studied and worked. He was killed by an IRA bomb. Now, it says, son of a research worker in tropical diseases. Now, Neil Hamilton Fairley was a little bit more than a research worker in tropical diseases. He was a brigadier in the Australian Army. He was in charge. He was the director of medicine, in fact, um, of the Australian Army, and involved in planning the Battle of Greece. That's in World War II. He had actually joined the Australian Army in World War One, immediately after his graduation. So, yeah, there's a little bit more to Neil Hamilton Fairley than being a research worker in tropical diseases. This is where it starts moving into the area of biological warfare, because Neil Hamilton Fairley, obviously, as the boss of the Australian Medical Corps during the Second World War, knew about biological warfare, and indeed the Bombay Bacteriological Laboratory was a biological warfare laboratory. Now, what the page on Neil Hamilton Fairley, which I should be able to find without too much difficulty. That's Neil Hamilton Fairley, and this is the Wikipedia page on him. He is with all his medals. Brigadier Neil Hamilton Fairley was an Australian physician, medical scientist, and army officer who was instrumental in saving thousands of Allied lives from malaria and other diseases. Now, this is a very uncritical review of Neil Hamilton Fairley, but it does say here that after the war he became consulting physician to the Hospital for Tropical Diseases and the Wellcome Professor of Tropical Medicine at the London School of Hygiene and Tropical Medicine. Now, if you look at what he did during the here at the LHQ Medical Research Unit. Now, this is actually coming from a book that was written in 2003, which doesn't tell the whole story by any means. But it says that Fairley travelled to New Guinea at the end of June 1943 and arranged for Plasmodium falciparum, which is falciparum malaria, cases to be evacuated to Cairns for treatment. At the time, as the flight time from Port Moresby to Cairns was only a few hours, this was considered safe. But since the disease can be fatal if not treated promptly, Fairley was concerned lest the cases be delayed for some reason. Movement control suggested that a special priority be allocated to such cases, and Major General Frank Berryman suggested calling it Priority Neil after Fairley himself. The LHQ Medical Research Unit used test subjects, all volunteers drawn from the Australian Army, including a small but notable group of Danira boys, brackets Jewish refugees, from the 8th Employment Company. Now, the news story that broke this in Melbourne, in the Fairfax News, around 1999 or 2000, said that there were 3 groups of subjects, what they call human guinea pigs in this new story. And they were um, disabled soldiers who were trawled from the military hospitals, Jewish refugees, as they have mentioned here, and interned Italians. Now that's an important omission. Interned Italians. And they were infected, it says here, infected with strains of malaria from infected mosquitoes or from the bites of other, or from the blood of other test subjects, which was then treated with various drugs. The volunteers were rewarded with 3 weeks' leave and a certificate of appreciation signed by General Blamey. The LHQ Medical Research Unit researched quinine, sulfonamides, atabrine, plasmaquine, and paludrin. Now, the newspaper story mentioned another drug, and that is paracetamol. And it said that Winthrop Pharmaceuticals, because paracetamol is produced by an American company, Winthrop, that paracetamol was, the trials continued after the end of the Second World War, through till 1946 under pressure from the drug companies, two drug companies in particular. The Winthrop and Imperial Chemical Industries, which were the company that produced this drug, paludrin, which was a German invention for malaria. And they were testing out paludrin. Now, the other thing that the Wikipedia doesn't say is where they got the malaria from. And the answer is the Red Cross. The Red Cross provided bags of blood that were infected with malaria parasites. The other thing is the danger of transfusing infected blood from one person to another. And the third very important omission is the CSIRs, the Committee for Scientific and Industrial Research, which became the CSIRO, or the Commonwealth Scientific and Industrial Research Organization. They had a mosquito breeding program where they had mosquitoes that carried thousands of times the normal load of malaria parasites. Now, obviously, if you released those mosquitoes into the world, they would cause a terrible spread of malaria. And that's what the CSIRO or the CSIR was doing during the Second World War. It's called entomological warfare, using entomological knowledge. It's a type of biological warfare. So this is what is not mentioned on Wikipedia regarding the malaria testing. Now this indicates that Australia played a key role in biological warfare during the Second World War, and the other evidence that I've provided shows that Australia has played a key role in the development and use of biological weapons after the Second World War, throughout the Cold War. And Neil Hamilton Fairley, he died in 1966, so he died before my father got his research grant for the lab. But you could say all these people hobnobbed together. So Neil Hamilton Fairley was a contemporary of Max Rosenheim and Graham Wilson. And my father's idea that somehow he was going to inherit John Walter's position just because he was his registrar shows that he had very little understanding of how the system works. You see, if you're not part of the powerful white man's club, and you're not born into it, you don't get in there.